Good afternoon, everybody, and thanks so much for joining the IGB Pioneer lecture for this semester. And it's really my honor and privilege to introduce Professor David Walt and to share with us things he's been doing in the area of diagnostics. I'll tell you that Professor Walt is someone whose career and accomplishments I've followed for many years, and in fact, teach a couple of technologies that he had a role in developing in my biosensor class. And so I hope a few students from my class find their way here today. Maybe they'll get some extra points on the next quiz. Dr. Walt is the Woese Professor of Bio-Inspired Engineering at Harvard Medical School and Professor of Pathology at Harvard Medical School and Brigham and Women's Hospital. He's a core faculty member of the Woese Institute at Harvard University and an associate member at the Broad Institute, and is also a Howard Hughes Medical Institute professor. Dr. Walt is co-director of the Mass General Brigham Center for COVID Innovation. So maybe we'll hear some things about novel COVID diagnostics. And also he's the scientific founder of Illumina and Quantarix, along with several other life science startups that he's been involved with in recent years. I won't read all their names because there's several of them. Dr. Walt has received numerous national and international awards. I'll just mention that he's a member of the National Academy of Engineering, a National Academy of Medicine, a fellow of the American Academy of Arts and Sciences, a fellow of the American Institute for Medical and Biological Engineering, and a fellow of the American Association for the Advancement of Science, and fellow of the National Academy of Inventors. And also was inducted into the U.S. National Inventors Hall of Fame. So it's really a privilege to have Dr. Walt with us here today. And Dr. Walt, the floor is yours, and welcome to Illinois. Thanks for coming to visit us virtually.
Well, thank you for the very kind introduction. And I'm sorry that I can't visit Champaign, Urbana today. I've visited the chemistry department once and also the Beckman Institute maybe 10 years ago or so. So would have been nice to get an update and see the campus. And I'm sorry for Illinois' loss last night in basketball. I'm a Michigan alum, so at least we've got one team representing the Big Ten still, but would have been nice if Illinois was made it further. I also want to say hello to a number of my colleagues who I recognize from Illinois on the call today. So thanks for joining. So I'll be telling you about ultrasensitive protein measurements. I know this is the Carl Woese Institute for Genomic Biology, but I think just in speaking with some of the faculty today, I think that's being interpreted appropriately, liberally. So my disclosures, really, this is required as a member of the faculty of the medical school. I'm obligated to show this. The only one that's really relevant today is as Dr. Cunningham mentioned, the one with Quantarix. I'll talk about the technology that was developed in my laboratory and then commercialized. I'll show you some of the clinical work that we're doing with that technology today. And as was intimated, you'll hear about some COVID work that we've both published and also some unpublished work. Let's just get right into it with single molecule detection. The first report of measuring a single molecule turned out to be a long time ago. It was 1961 by a Stanford faculty member, Boris Rotman. And what he did was he just made a water and oil emulsion with a fluorogenic dye or a chromogenic dye at the time, I think, and distributed a low concentration of an enzyme, beta-galactosidase, in this water and oil emulsion, and saw that some of the droplets turned red and other ones remained colorless. And his conclusion from this was that he had isolated individual molecules of the enzyme, which in the presence of the substrate would catalyze local concentrations of this dye. And that's what he was measuring. And so, this is actually remarkable. 1961 was really the first single molecule experiment. We, probably 25 years ago, had discovered that we could create microwells on the end of an optical fiber. And I'm not going to show you much about this today, but suffice it to say that when we started doing our single molecule work, we had some of these microwell arrays on the ends of optical fibers lying around in the laboratory, and I had an idea at a lab meeting one night that, look, if we look at the volume of one of these microwells, it's only 40 times 10 to the minus 15th liters, 40 femtoliters, and you don't need that many molecules in a very tiny volume to give you a locally high concentration. And so we proceeded to do a very simple experiment, which was to take this optical fiber array, translate it through a droplet of solution that contained an enzyme at very low concentration, same as what Rotman had done, this fluorogenic substrate, and just took that. There's 60,000 microwells on the end of this optical fiber array, pressed it through, sealed it against a PDMS membrane, which sealed each of those individual 60,000 tiny femtowells from one another. So if there was an enzyme trapped in one of those microwells, it would generate a locally high concentration of fluorescence, which we would be able to detect, and the wells that contain no enzyme would remain dark and colorless. And so a student, Dave Risson, who was a graduate student at the time, carried out this experiment, and here's what he got. It was, he brought these results to my office. Background fluorescence was non-existent in the absence of an enzyme. In the presence of a relatively high concentration of enzyme, we had roughly one enzyme for every 5 wells. And as we diluted, it went down, and you could literally count the number of active wells. And so we published this, I think it was in Nano Letters at the time. And what we claimed was that this was really the first time that you could measure the concentration of something by simply counting the number of active wells. And so this was the predicted number. Here were the 3 replicate experiments, and here's the average. And we got good agreement between what was expected from the calculation based on the concentration of active wells and what we actually got experimentally. But of course, who cares about measuring the concentration of enzymes? I wanted to throw this slide in because I think it's important to sort of set the stage. I know this is the Institute for Genobiology, but of course, genomics, which I've been involved with extensively, measures propensity. That's the genotype, but of course, proteins measure phenotype. It's closer to the biology. And so, there's great methods for measuring DNA and RNA, inexpensive, high throughput, accurate, sensitive. We've got, amplification methods, but proteins have traditionally been a challenge simply because of the technology that's able to measure them. And so let me now turn my attention to really talking about how we address this problem. And as we were thinking about this, just in terms of framing, why do you want to measure proteins? Well, if you're interested in health, then there are 325 proteins that exist in the serum between millimolar and picomolar. So 10 to the minus 3rd molar to 10 to the minus 12th molar. Millimolar, things like albumin, which is just in very high concentration. Picomolar are things like cytokines, TNF alpha, some of the interleukins. So of those 325 proteins that are known to be in that concentration range, about half of those turn out to have some sort of clinical utility. And so we said, well, what's preventing things from being measured at that low limit of detection? Well, it's this very nice but now 40-year-old technology called ELISA, enzyme-linked immunosorbent assay. And for those of you who aren't experts in this, I'll show you what that is in a minute. But the question becomes, how many proteins exist below that surface that we're able to measure? And we know that there are a little over 20,000 proteins in the human proteome. These are just the open reading frames, the expressed genes in the human genome, and it's calculated, and this was done, by others, that about 4,000 of those, or slightly more, should appear in the serum, but over a wide concentration range. And so we said, is there a possibility for being able to measure these, and now opening up a different insight into what had previously been uncharted territory. And so this is sort of the value proposition here. It's really moving away from sort of the traditional technology, which is typically done in microtiter plates, where an increasing signal, that is, it could be a intensity of color, it could be the intensity of fluorescence, correlates with increasing concentration. Of course, this requires calibration, because if I said, what's the concentration here, you'd have to have some way of comparing that green dot with another green dot that had been done using some standards. Whereas if you're talking about single molecules, now if I say, what's the concentration here, or make it even simpler, how many molecules are here? Well, it's easy to see. There's 4 molecules that are appearing in this field of view, and there's more here, and there's 1 here. So it's an absolute measurement. And I've argued both in literature, and I'll repeat it here, is that if I give you a volume of solution, I challenge any of you to figure out a better way to tell me what the concentration is than to tell me how many molecules of a particular protein or a particular small molecule there are contained in that volume. If you know what the volume is, the number of molecules, then it's easy to calculate the concentration. And again, it's an absolute measurement in principle. So how do we accomplish this? Well, we call these single molecule arrays, or my lab coined this term SIMOA for short. And the idea is identical to an ELISA, a standard ELISA technology. It just digitizes it. You've heard digital PCR. Digital PCR takes a large solution of a high concentration of nucleic acids and digitizes them by partitioning those molecules into droplets and then running the PCR reaction in those tiny droplets, similar to what Rotman did with beta-galactosidase in 1961. We took a slightly different approach, and it turns out to be a little bit more challenging than digital PCR for the following reason, is that when you get down to incredibly low concentrations of molecules, and those chemists in the audience understand this. This is what Rob Korn from UC Irvine calls the tyranny of the Langmuir binding isotherm. When you get down to really low concentrations, then mass transport to a surface becomes rate limiting. That is, binding to the surface is rate limiting because the diffusion of those very small number of molecules to that surface is so slow that it can take months, if not years, at certain concentrations and certain sizes of molecules for that solution to come to equilibrium. So what we did is we said, well, why don't we use beads to bring the surface to the molecules instead of trying to bring the molecules to the surface? And that really was sort of what prompted this whole approach. So I've got a touchy mouse here. So the idea is we take lots of beads, 500,000 beads, and just to give you an idea, in 100 microliters of let's say a biological sample like blood, if you have 1 femtomolar concentration of that protein, you can calculate this, just general chemistry, Avogadro's number. 1 femtomolar calculates to be 60,000 molecules. So that's an order of magnitude, that's 3 logs, actually 3 logs lower than picomolar concentration, which is the lower limit of detection for the traditional ELISA. So you have to add 3 zeros to that. The lower limit of detection for a traditional ELISA is 60 million molecules. That's the best you can do in 100 microliters of a blood sample. So we said, how would we detect 3 logs lower than that, 60,000 molecules? So the way we do it is we take antibody coated beads. These beads are on the order of about 2.5 microns in diameter. We've titrated this a long time ago. About 250,000 antibody molecules are able to pack onto an individual bead. So you take 500,000 of those, which is almost 10 times as many beads as there are molecules in the solution, with each of those having 250,000 capture antibodies on it. So Le Chatelier's principle, we just suck all the molecules out of solution as a consequence of providing a huge number of receptors in there that can capture those molecules, and a huge surface area that allows diffusion to take place quite quickly in terms of the number of beads hitting the number of molecules. And so we get very rapid equilibration of binding. We then do exactly what's done with a traditional ELISA. We tag that with a detector antibody that recognizes a different epitope on the same protein, so just a slightly different, site on the protein. That detector has a biotin attached to it, and then we label that with a streptavidin labeled beta-galactosidase, the same label that I showed you previously with that single molecule enzymology experiment. And now, if there is an enzyme on this bead, it is able to catalyze the formation of many molecules of this fluorescent dye per second. The turnover number of beta gal is about 400. So we take those beads, we then put them into the microwells. The microwells are able to accommodate a single bead. So we etch these wells so that they're deep enough to only contain a single bead. Their diameter is about 3 microns, so there's size exclusion. We then seal it with a perfluorinated oil, and along with substrate. And then what happens is, if there's an enzyme that is associated with a bound protein molecule, those protein molecules now light up as a consequence of the enzymatic activity that has labeled them. And so that allows us to do a digital assay. So the first thing that we did, this was published about a little over, it was about 10 years ago in Nature Biotechnology, was we measured PSA. So it was just an assay that was, we had antibodies available, good binding antibodies, and the limit of detection of sort of the traditional ultrasensitive PSA, and I'll tell you what it's good for in a few minutes, is 10 picograms per mil, which is here. And we started running our assay, and we saw, wow, there's a bunch of data bunched up here. Let's expand it. Still a bunch of data bunched up here. And finally we get down here. and so our limit of detection using the same antibody binding pairs as ELISA, but now digitizing the signal, allows us to get about 3,000 times more sensitivity, analytical sensitivity, compared to the current state-of-the-art ultrasensitive methods. Now, I'm not going to talk a lot about technology today. I'll talk a little bit at the end towards sort of our next generation version of single molecule detection. But for now, we published this last year in JAX with multiplex detection of simultaneously small molecules, nucleic acids, and proteins. And here's some graphs from the paper. We did a cortisol assay, which is small molecule IL-6, which is the protein, and we did a microRNA assay, all simultaneously, just with different kinds of capture beads. And you can see that even the threeplex, the multiplex assay, is almost indistinguishable from the singleplex, and everything works well. If you're interested in this, go read the paper. As was mentioned earlier, this technology has been commercialized. This is a company that was founded in 2007. It's now a public company called Quantarix. They make multiple products that employ this, the underlying technology that was licensed from my laboratory. This is something called the HD. I guess this is the HDX. I think this is the HDX. It's essentially a refrigerated, automated immunoassay system. It has robotics in it. It can run 400 samples in 5 hours. So you load 4 96 well plates. This one is the more manual version, which requires a plate washer, but it can do a 96 well plate manually, and then you just put the plate in, and it does all the other stuff automatically in terms of loading the beads and everything else. This is what the microarrays look like today. For the automated instrument, what was designed were microwell arrays on the end of these little fluidic chambers right here. So beads are loaded into these microwells. You can see, I think I have a better picture here. Yeah. So there's 216,000 microwells in each one of these arrays. At the end of one of these, this is the top, this is the top view. So the beads are loaded in here. They get magnetically loaded into the wells. They get loaded and sealed with substrate. Perfluorinated oil seals them, and then waste goes out. So 24 multiplex assays can be run on a disk. I'm sure you recognize these disks. They're what used to be used for DVDs and Blu-rays. Most of that business has dried up, and the companies like Sony have now migrated to making plastic consumables that fulfill the life sciences market. So this was actually one of the first uses of this technology. So let me spend some time talking about the clinical applications, and I'll go through some of this pretty quickly, and then we'll get into COVID. But I want to illustrate a few, I think, themes here, because I think it's important as we go that I describe at least my perspective on how I think personalized medicine and personalized health will benefit from some of these kinds of technologies. So the first application, as I mentioned, of the Simoa technology was doing prostate cancer surveillance. And this is not to do early detection of prostate cancer. This was a very specific application. As many of the 50-year and older men know, PSA is sort of used as a marker for, it's a specific marker called prostate-specific antigen. That's what PSA stands for. It's a protein that's produced by the prostate gland. And generally, higher levels of PSA in elderly men indicate that there may be prostate cancer. And if somebody ends up having a high PSA, they get a biopsy. If the biopsy turns out to be positive, depending on the person's age, they may decide, look, it's a slow-growing cancer. And in fact, most men will die with prostate cancer, not of prostate cancer, just because it tends to be a slowly developing cancer. But if a person's young when they got diagnosed, or it's a more aggressive form of prostate cancer, then they'll do a surgical resection. That is, they'll take out the prostate. And what happens is, because it's a very specific marker, that protein has a short half-life. It will plummet in the first few months after surgery, and it will go to non-detectable levels, okay? So generally, they leave some tissue, some prostate tissue in the patient, because if they don't, it leads to incontinence, it leads to certain sexual impotence. And so they tend to take as little tissue out as possible, but still capture the margins such that they take out all the cancer. But in some cases, that residual tumor was left. They could not fully resect it because the tumor was either too large or was heterogeneous, or the tumor had metastasized prior to the surgery. And so men who have prostate cancer surgery, called a radical prostatectomy, removal of the prostate, oftentimes will recur. And the recurrence occurs as a result of that residual cancer growing over time. But these men come in every 6 months, get a blood test, and the blood test, in most cases, just says non-detectable PSA, because the blood test is not sensitive enough to quantify or detect the low levels of the residual tissue produced PSA. At some point, if their cancer grows and returns, then what happens is they start having a measurable level of PSA, and their urologist tells them, well, I've got bad news for you. You have molecular or biochemical recurrence, meaning we can now measure PSA level, which indicates that there is likely to be the presence of another tumor. So, or the growth of the existing tumor. So what we did is we took samples from men who had been 5 years post-radical prostatectomy and had undetectable PSA levels. And we measured their blood. And we could quantify every single one of them with that ultrasensitive PSA test that I showed you previously. Here is the red dotted line is our limit of detection. The blue line right here is the ultrasensitive test, which is only used for measuring men who have had prostate surgery. And the green is the sort of limit of detection of the standard PSA test that's just used for screening purposes. And as you can see up here, these are sort of the normal ranges up here, way up here, of the typical PSA levels in normal healthy individuals. So we could quantify every one of these men. And what's interesting is they vary by 3 logs in concentration. I'm not going to make any claims about the person at the high end here compared to the low end. They were all considered to be cured because they had 5-year non-recurrence. So what we did is we got our hands on a set of samples that had been collected. This is a retrospective set of samples from men that had had prostate surgery and they had serial samples taken every year or every 6 months. And what we did is we just looked at those individuals, and you can see that this group of individuals after 5 years were only 35% cancer-free. But what it turned out was that the way that we could differentiate the non-recurrers from the individuals who were at high risk were if we could measure at a time point between 3 and 6 months post-surgery, a single time point, and their concentration was less than 3 picograms per mil at that time point, then 100% of those individuals did not have recurrence after 5 years. If, on the other hand, they were greater than or equal to 3 picograms per mil, only 35% of them were cancer-free. Now, this is still, I think, less than ideal. I mean, it's great if you're in this group because, all the psychological trauma associated with coming in every 6 months and not knowing about your prostate cancer status sort of goes away, because even after 6 months, we can say, look, go home, don't worry about it. You don't have to think about it for at least 5 years versus this group. It's still less than ideal. And so right now, there are some clinical studies with this technology that are, instead of looking at a single point, they're looking at the longitudinal sampling. That is, if the PSA level is stable over time, then the conclusion, we think, is that there's no active disease. But if there is an increase in the level of PSA over time, that is the rate at which that increases, if it's non-zero, then that's indicative of recurrent disease. Okay, so I'm going to turn my attention to something that's more complex, because PSA, I think, is almost a unique case. It's unique in terms of its presence in a single organ, the prostate gland, and it appears both in healthy individuals as well as cancer individuals, and it can only be used, in that very, I think, selective kind of scenario that I just described. But we think that almost all cancers secrete biomarkers into the bloodstream, and those can be those can serve as markers of cancer. And that furthermore, it's not just a single marker, but it's a pattern of these markers that are going to be diagnostic of disease. Okay, so first of all, I'm just going to cut to the chase here. We did some animal studies. We actually used a prostate cancer cell line where we continued to dilute and inject in a mouse xenograft model a human prostate cell line. We ended up being able to detect PSA at a level that would correlate to a tumor in a human that would be 1 millimeter in diameter. So that's, sort of back in the envelope calculations based on some assumptions about secretion, and number of molecules, and number of cells, and doubling times and all that. But that compares with the best imaging technology today to be able to detect a solid tumor is about 0.5 centimeter. So if you do the math, this is 125 times smaller volume, which would correlate with a much earlier stage of cancer in most cases. So I'm going to talk about breast cancer. We know that this is, remains a high cause of cancer for women. In 2008, there were more than 270,000 new cases, 41,000 deaths. We also know, not only from breast cancer, but from all cancers, that early stage cancer has much better survival rate than later stage cancer, where it's metastasized and had a chance to incorporate many more mutations than in a localized early stage cancer. We also know that the present way in which breast cancer is diagnosed is highly imperfect. What happens is that with mammography, there are many false positives. After all, it's not looking at biology, it's just looking at an image. And that image oftentimes gives a false positive. Everyone that has a false positive on the mammogram, meaning that there is some anomaly on the mammogram, has to go in for some sort of needle aspirate biopsy, which is invasive. And of those biopsies, I mean, this is pretty staggering. 70% of them are negative. So, the sense that we were talking about, or, we've heard in the COVID era, 99% sensitivity, 99% specificity. This is after the mammogram, after the biopsy, 70% of those are negative. So there's lots of unnecessary biopsies, more than twice as many unnecessary ones as necessary ones. And so, huge false positive rate leading to the biopsies, and they also will miss cancers that just because they're too, it's an imperfect method. As I said, it's an imaging technology that can only detect something at the smallest, that's about a half a centimeter. So our approach is, if there's a solid tumor here, it's secreted into the bloodstream, and our hope is that we can detect some of those biomarkers. And so we talk to lots of key opinion leaders in breast cancer oncology. We canvass the literature. We ask them, we ask people, what marker would you measure if you could measure it? And, some of this is based on immunohistochemistry markers that are used in pathology. But, and this is, you're going to see this a few times over the next few minutes. Here are cancer patients. Here are healthy patients. Here are cancer patients. Here's healthy patients. There's a few hundred dots under each of these populations. It's very hard to differentiate these individuals just by looking at a single marker. You might say, well, this person, and of course this is a log scale, this person has, is an outlier relative to all the other normals, but that's 1 out of a few hundred. So that's not, going to do it. That's not a great marker for cancer. But if you now take all these markers and use a classification algorithm that is, trained either with, depending on how many subjects you have, a variety of different classification algorithms. Could be, a machine learning method. It could be, just a supervised training with, some regression analysis. The 24 markers, that is the relationship between those markers in cancer patients, is very different from how it presents with healthy individuals. And in fact, we have, the ability to, we have an accuracy on the order of 95% or better for just being able to detect breast cancer versus non-breast cancer. Now, all these individuals I should mention, I neglected to say how we collected these samples. These were all collected from individuals who had positive mammogram, positive needle biopsy, but were naive to treatment. So they had not had surgery, they had not had any sort of chemotherapy, they had not had any radiation. So these were treatment naive individuals, so no intervention. And even with only 4 biomarkers, we had the ability to be sort of in the high 80s. Okay, so that's, that's sort of one thing. I should also mention that it was incredibly important to include the woman's age in this algorithm. And the reason for that is that some of these markers, so you can see here are healthy patients. There's just a natural increase over time. That is, the levels of the concentrations of this marker GDF15 increases simply as a consequence of a woman's age, whereas other markers like CA 15.3 do not. They're just flat over time. And so by accounting for the woman's age, we are able to accommodate this. Okay, we've now gone on. I'm not going to belabor this, but now what we're trying to do is really subtype these individuals. So when a woman gets diagnosed, it's not just yes, you have cancer, no, you don't have cancer. It's you have estrogen receptor positive or HER2 positive, which is going to be treatable by Herceptin, or a hormone therapy, or triple negative, which is the most aggressive form of breast cancer. And so by now subdividing some of these cases by subtype, we're now beginning to be able to do with a blood test what pathology is able to do. Again, all this is unpublished, it has not been peer reviewed, but I thought I'd give you an early peek at some of the results coming out of the lab now. And we've done a lot of analysis on this. So I want to get to the COVID stuff. One other thing I just want to mention, and I think I alluded to this, you could sort of see it with the breast cancer subjects, but we did a study a number of years ago with some college students where the purpose of the study was really to look at the effect of the seasonal flu shot on the host response. That is, looking at certain protein markers that either could predict a vaccine was effective or not effective. What turned out to be the most interesting outcome from this study was that we found that if we measured these 15 cytokines, this is a particular volunteer, one of the participants, again, a 20, 21-year-old college student. This particular individual, their cytokines remained incredibly stable over 90 days. This person, same thing. This person's cytokines are close to the limit of detection. That's why there's a little bit of noise down here. But if you now plot, something like IL-10 with 5 individuals, they're pretty well controlled within, maybe a factor of 3 or 4 of each other. But if you look at something like IL-8, they vary by at least 2 logs. And when we went to many more individuals and started looking at these markers, both the intrasubject variation, that is how closely controlled these markers are within a person over time versus how much they vary between individuals. That's what the white bar is. The black bar is within an individual, very tightly controlled within people. But many of these markers, IL-8, for example, we've done lots of studies now and have shown that your concentration can vary by 4 logs between ostensibly healthy individuals. So what is baseline for you may not be baseline for somebody else. And so we think this is going to be incredibly important because it's not just your instantaneous concentration of some of these markers. It's sort of what's your baseline supposed to be. Okay, so I'm going to transition to COVID now. My lab, we pivoted within about 2 weeks after the first case of COVID in Boston. You all recall the Biogen conference was here. Boston was one of the first places after Seattle that began to have effects of the pandemic. And so rather than shut down the lab, about half of the group decided that they wanted to work on COVID. So we developed one of the first antibody tests. We took 4 color-coded beads. We put spike protein, nucleocapsid protein, the S1 protein, which is a subunit of spike, and the RBD, the receptor binding domain, which is a subunit also of both spike and the S1. This is the binding domain that's responsible for engaging with the ACE2 receptor on the host cell. We put those on different beads. And then what we did is we ran assays where we took those 4 beads, incubated them with patients' blood serum, and then interrogated the IgG fraction, the IgM fraction, the IgA fraction. IgM are the sort of immature antibodies. The IgA fraction are mucosal antibodies. And we thought this was going to be important because it's a lung disease and it infects the mucosal lining of the lungs. And then IgG as well as sort of the mature antibodies. And so we were able to develop this in very short order. Frankly, it was because some of my colleagues at Harvard Medical School had already generated some good binding antibodies and also had expressed some of the proteins before others were commercially available. So we have the high sensitivity, we have high resolution because we're measuring 12 different interactions, all 3 IgGs with all 4 proteins. We have wide dynamic range, and we also have the ability to multiplex. And so, we developed the assay. This was published in Nature Biomedical Engineering pre-pandemic. There's clearly some reactivity here with some of these individuals, probably from other coronavirus spike proteins. These are PCR negative. We know at the time there were lots of challenges with PCR. They were giving false negatives. Things have subsequently been corrected, but we were pretty sure that we were detecting real positives, even though they were PCR negative. And then you can see that as COVID develops over, and these were in patients 0 to 3 days, 4 to 7 days after admission to the hospital. And over time, you can see that everybody seroconverts except for one individual who happened to be immunocompromised. Again, I don't want to overwhelm you with data, but all the data from lots of patients are shown here. Red are shown immunocompromised individuals. You can see they have a lower seroconversion. That is, they don't develop as strong of an antibody response as others. But I think what turned out to be incredibly important was our ability to look at over the entire range of antibodies and antigen interactions, the development of antibodies in different patients. And so this patient, for example, within a few days of admission to the hospital, seroconverted, started to develop antibodies, first IgM, then IgA, and then IgG, and eventually developed very strong antibodies to the virus, whereas an individual who turned out to not survive infection never mounted an immune response. Again, I want to get to some of the newer stuff in the last few minutes. So, we had excellent sensitivity and specificity with respect to, creating very early detection of seroconversion in patients. And we're now monitoring out over, this was just over 90 days, but, we can monitor the individuals and look at the strength of their residual immune response, months after their infection. So I just want to get to 2 other topics that relate to the viral antigen. The first is that we started, back in August, and this was published in Clinical Chemistry in 2020. So we developed an ultrasensitive antigen assay, to S1, to the full spike protein, and also to the nucleocapsid protein, which is inside the virus. What you can see is that we get, and so this is actually in blood. So this is the first measurements. I think these are still the first and only measurements that have ever been made of the viral antigen appearing in blood. And what turned out was that we could differentiate not only between the negative patients and the positive patients, but more importantly, we could monitor both the efficacy of the immune response in individuals to clear the viral antigen. And so what you can see is that after they came in, had a first PCR test, this is after day 1, by day 4, this individual had already started to mount an immune response and had eliminated the antigen from their blood by day 5. I think and I'm going to skip ahead here, this is sort of a heat map, but we're showing 39 patients. Here's the levels of antigens here. Here's the level of antibodies. And over time in these individuals, you can see that the antigen goes down and antibodies go up, and these are all individuals horizontally here. So I think what was even more striking was the fact that the presence of antigen in the blood when they appeared in the hospital was highly correlated with their outcome. That is, if we could not measure antigen in their blood when they came into the hospital, then they actually had either no need to go to the intensive care unit and be intubated, or they had a much longer period of time before they ended up in the ICU. Whereas individuals who had high levels of antigen, and when I say high levels of antigen, I mean like femtomolar concentration of antigen in their bloodstream, those individuals invariably immediately went into the intensive care unit and needed to be put on a respirator the day that they showed up in the hospital. And so these are actually being used clinically now. We're taking patients who present in the emergency room, come in, they get an antigen test, blood antigen test, and this helps advise the clinicians about what the course of their infection is going to be. The last thing I want to talk about, which is incredibly exciting work. This should appear soon. I can't say where at the moment, but we actually looked at the Moderna vaccine. We looked at patients who had received the vaccine. This is the 2-dose vaccine. We know there's been some recent reports, showing the antibodies, the serology after the first dose and the second dose. I want to show you what happens here, because with this vaccine, the thought is that all this stuff happens in the lymph nodes. This is what's expected, that there's no antigen that gets out into the system. Everything is localized and gets in the lymph nodes and is produced in the lymph nodes. Well, we've shown that to be, it turns out that that's not the case. So we did the following experiment. We took a high sampling of individuals before they were vaccinated, 1 day after vaccination, 3, 5, 7, a week after vaccination, 4 weeks after vaccination, but right before their second dose, and then days 1, 3, 5, 7, out to day 56, okay? And we measured both their antibodies and antigens, and lo and behold, we found that we could begin to detect antigen in the bloodstream of these individuals. So the mechanism of this messenger RNA vaccine is that it's supposed to get into cells, gets translated into protein in the lymph node, but the fact is that some of that protein is actually leaking into the bloodstream and is getting into and is systemic. And you can see that it sort of peaks, and then after 9 days, it's undetectable because 100% of these individuals have seroconverted, after day 28, it's then a new injection of the vaccine, but it never goes up above the limit of detection of the assay. We do the same thing with the spike protein. We see a few examples where the full spike appears, but nucleocapsid, which is not part of the vaccine, that's that internal protein in the virus, that's not part of the vaccine. The mRNA vaccine is only spike protein. This is a great internal control. We never detect any nucleocapsid. And I think what's even more interesting is that we can measure these responses. So after 28 days, every one of these individuals has immunoglobulin to the S1, which is what you hope. And then within a couple of days after the second dose, they all have about 10 to 100-fold more immunoglobulin, more antibody than they did before their second dose. So the boost works, but I can assure you that after you get your second boost, I would say wait a week. You don't have to wait 2 weeks because there's nothing that happens after a few days. I'm going to stop here. We have some other technology that we've developed called Dropcast Simoa. It was just published and is really a way that we're now using this same single molecule technology to go to much more sensitive measurements, but is amenable to a point of care kind of application. So I'm just going to skip here. These are the folks who were involved in the postdocs and graduate students who were responsible for the COVID work in my lab, and lots of collaborators on the clinical side. And then some of the other work that I talked about earlier on the breast cancer and the technology development right here. And then this was the last shot pre-COVID at a lab meeting. We do not have, I guess I could show the virtual Zoom group, but I think this is what people want to see and what we want to get back to. So thanks again for the invitation. And I know I've left just a couple minutes for questions, but happy to answer them.
Thank you very much. Thanks so much, Dr. Walt. So we do have time for some questions. I'll look at the chat, but otherwise you can unmute yourself and address your question to Dr. Walt directly.
I don't see any questions in the chat.
I'm looking there too. So I'll ask the question to get things started. So first, congratulations, really exciting results across the board. I'm curious, just going back to the prostate cancer example you gave, it seems like very thorough clinical validation for that more sensitive type of test. What's the roadblock for getting that into regular clinical practice as just part of treatment? I'm a man over 50, so I'm wondering when I'll use a test like that for detecting my PSA.
Well, I think it's just the way that clinicians have been trained conventionally is single time point, and there's a normal range, right? And that's glucose, cholesterol, all the small molecules. We know that there's normal levels, and I think that extends to some of the higher concentration levels of proteins. It's like some of your liver proteins and like alk-phos and things. But I think what is really the barrier is just this sort of dogma that there must be a normal concentration range for every person, and that every person should fit into that population average. And I think as we begin to look at these lower concentration proteins, we see that there's incredible variability. I think that the breast cancer slide that I showed earlier was a great example where just eyeballing it, you couldn't really tell the difference between the healthy controls and the cancer patients, but certainly the relationship between those markers is informative. And so I think it's just I agree with you. I think it's just getting getting people to think differently. But we're working with a couple of urologists at some of the top research, cancer research centers, and they're believers at this point. And I think it's just going to take a few papers showing that you want to measure longitudinally as opposed to a single time point.
There's a question in the chat from Lori Leonelli asking if about the background binding, if that ever creates like a noise floor for like non-specific signals.
Yeah, Lori, that's a fantastic question. And it is really the reason that ELISAs turn out to have the relatively high limit of detection that they do. And that's because, first of all, think about, picomolar concentration, 100 microliters, as I said, 60 million molecules. You have to get 60 million molecules onto a surface, which is typically a polystyrene microtiter plate. You need a very high concentration of labeling reagents in order to effectively label the molecules that have been captured on the plate. And that will invariably lead to non-specific binding. So just as you suggest. So what we do with our beads is, because we only have a few molecules that we need to label in that solution, we can use first many orders of magnitude lower concentrations of the labeling reagents, so they don't stick to the surface in the first place. And two, because they're beads, we can wash them much more effectively than you can a solid surface, which requires agitation. So really good point. And we still have non-specific binding. Different proteins, different antibodies will give us more non-specific binding than others, and that will give us our floor of what our limit of detection is for a particular assay. And those can vary by a factor of 10 or a factor of 20 in some cases, if you have sticky antibodies, sticky capture antibodies versus antibodies that really only capture the molecule of interest. So great question.
Perhaps one more question. I want to make sure people get an opportunity that have been patient. Just unmute yourself and speak it out there.
Looks like
this is Xing, David. This is a nice talk. This is Xing. Sorry, I jumped in a little bit late. So for your earlier antibody and antigen test on COVID, can you comment on the cross-reactivities for specificity? I know you mentioned that for the cancer one, but.
Yeah, so we definitely see some cross-reactivities with some of the spike proteins, most likely from prior or other coronaviruses. We are now actually evaluating other antibodies with the idea that if you use multiple antibodies against the coronavirus spike, you'll get a unique sort of ratio. And if you can use that ratio to determine whether it's specific binding or non-specific binding to the SARS-CoV-2 spike. And then we now have, again, another whole effort now to update our antigen assays with some of the variants. We've gotten our hands on some of the proteins expressed in recombinantly from some of the variants that allow us to now make a multiplexed assay that will allow us to look for some of the variants using an antigen assay instead of doing it with PCR.
Okay, thanks. So I'm just interested in those molecules, and maybe at some point Brian E also in the audience, and we might be interested in trying those antibodies.
You have my email address, so
yes. Okay. Thank you.
Sure.
Well, thank you very much. Oh, I see one question. He-sun, maybe like one more question before we finish for this afternoon. He-sun?
Yeah, thank you for the great talk. I was wondering, because different patients have different lipids level, so their non-specific binding might be a bit different across patient as well. Do you dilute your sample to avoid non-specific binding, or do you do like extra tests just to see? Because we also try to do some single DNA detection or RNA detection. And if it goes down to the digital, right, every single non-specific binding counts. So I was wondering if there's any patient dependency there.
Yeah. So dilution, it turns out that dilution is important. I didn't even mention this, but our antibody assays, we are diluting 1 microliter of blood 4,000-fold. So, at that level, any non-specific binding is not going to be measurable. Now, that's not for all assays, right? But we typically, will dilute to ensure. I mean, that's the advantage of ultrasensitivity, is that you have the ability to dilute and sort of wash out things that could, create non-specific binding. But I'll tell you, it's really a big issue. I mean, there's a lot of fans of this technology called, the SOMAmer technology with SomaLogic. And, we've had multiple collaborators who have come to us having run SomaScan, and they've said, oh, look, these 6 proteins are really indicative of disease X, Y, or Z. And then we create specific assays, and we cannot recapitulate any of the results that they got on SomaScan, because if you think about it, you have 4,000 different binders that are all reacting a little bit, even a very small amount with, all the thousands of proteins that are present in a sample. You're going to get cases where there's going to be some antibodies or some aptamers that are going to be sticky, and they're going to be reproducible, but they're not going to be, they're going to correlate, but they're not going to be causative, right? They're not going to tell you anything about the underlying, real biology of what's going on. So I think this is a huge area of worth investigating.
Thank you.
Well, thanks so much to everybody for coming. I know we lost some of our audience at the top of the hour. That's the way Zoom meetings are. But let's just thank our speaker again. Thank you very much for a fascinating talk. So if you could hear us clapping, there'd be clapping in the room.
I see all the virtual claps, so thank you.
Thank you.
Dr. Walt is going to meet in 12 minutes with grad students and postdocs. So thank you very much, David, for taking some time to chat with some of our aspiring, brilliant young people. And so thanks again so much for spending a big chunk of your day today with University of Illinois.
Great. Now I'll see you shortly.